So today we are observing an onion scale and we are going to identify onion cells. Before we begin, we want to remember to rotate the nose piece so that the shortest objective is pointing down, and we want to make sure that the stage is as far down as it can go. We do that by rotating the coarse focus knob. All right, so we need to get our scale. Scale is part of the onion bulb, and we're going to remove the epidermis. So it looks like this. On the inside, there's just this really thin layer that we're going to take off with our nails. And we've got this really thin layer. We're going to add a drop of water to our microscope slide, and we're going to place the layer of the epidermis into that drop of water. We want it to be flat, not folded. Then we're going to add 1 drop of methylene blue on top of the onion tissue. This is going to make it easier for us to see organelles inside of the cell. I'm going to try that one more time. So I started with another one because I thought I could do it better. What we're doing right now is we're going to wait for a minute before we put the small cover slip back on top of the stained epidermis. So we put the cover slip at an angle and we drop it down. We are going to put a drop of water on the opposite side of the cover slip, and we're going to use some paper towel to draw the excess water and the stain from underneath. All right, and now our slide is ready to observe under a microscope. So we're starting with the lowest power objective. We're going to turn on our microscope. Go. So this is the lowest power objective. So those flattened rectangular discs are individual cells. See how they're all connected to each other to make up the actual onion scale. So this is looking through the medium powered objective. Noticed the dark structure in the center. That right there is the nucleus of the cell. It's been stained blue so that we can easily see it. So up at the top of the focus field of view, we can see 2 small dots, and so those are also nuclei. Now, somebody asked, why don't we see the nucleus in all of the cells? And so the central vacuole can be so filled with water and glucose that the nucleus is pushed all the way up against the cell wall, making it difficult for us to see. Something else to think about is that we might be looking at more than one layer of cells here, and this is why the cell looks to have more than one nucleus. So now we're looking at the field of view with the highest power objective, and I moved the diaphragm to decrease the amount of light that was coming in through the microscope slide. So what we're supposed to be able to observe here is the cell wall and the cell membrane. Remember that plant cells have a cell wall and a cell membrane, so you can kind of see two layers that are stacked on top of each other. So here I'm focused on the nucleus. It's pushed up right against the cell membrane and the cell wall. And we're just gonna look around here. There's another nucleus that's easily visible.